In this video, we are going to talk specifically about the trip and lac operons. Recall that an operon includes genes in a metabolic pathway, and these genes are called structural genes, and these genes are grouped together on a chromosome and typically transcribed at the same time. And remember that these operons are existing in prokaryotic cells, which, for example, includes bacteria. And these two different examples of operons. The first one, the TRP or trip operon, the TRP stands for tryptophan. And tryptophan is an essential amino acid. And what this means is that it is an amino acid that is essential for the survival of the cell. So if the cell does not have tryptophan, it needs to be able to make tryptophan. The lac stands for lactose. And lactose is the sugar found in milk. And the lac operon includes genes that code for enzymes that help the prokaryotic cell break down lactose. One reason why we are focusing on these two particular examples of operons is that the trip operon is a repressible operon. This means that the default is that it is on. And when we mean on, it means that gene expression in the form of transcription and translation is occurring. So the repressible nature of it means that the default is on, but it can be turned off when the cell does not need to make tryptophan. Now the lac operon is called an inducible operon. In an inducible operon, what this means is that the default setting is off, meaning that gene expression is not occurring unless it gets turned on. So first, let's take a look at the tryptophan operon or the trip operon. And if you haven't watched my other video about operons, I recommend that you go back and watch it because it will help you recognize this kind of simplistic drawing of what an operon is. So remember that an operon, we're looking at a strand of DNA that is in a chromosome, and there's an area where the regulator gene is, a promoter region where the RNA polymerase will attach to, an operator region where a repressor may or may not attach to, and then we have the structural genes. And in the case of tryptophan, these structural genes are coding for the production of enzymes that will make the amino acid tryptophan. Now, recall from the introduction slide that the trip operon is a repressible operon. This means that the default mode is going to be on, meaning the default is that these genes are going to be expressed, which means that transcription and translation is going to occur. And so what happens is the RNA polymerase is an enzyme and it binds to the promoter. And as long as nothing is preventing it from continuing along the strand of DNA, it is going to travel along the rest of the operon and it is going to transcribe these genes. And then the mRNA is going to go through translation in order to form the proteins that are going to be converted or made into these enzymes needed for the cell to produce tryptophan. So remember that this is a repressible, so the default is on, meaning gene expression is occurring as the default. Unless it is specifically turned off. And it would be turned off by a repressor molecule. And remember that gene expression just means that transcription and translation are occurring so that a protein is being made. Now, if the cell has tryptophan present, so if it doesn't need to make tryptophan, it can turn this process off because remember that making these enzymes, and in this case, making the tryptophan, it requires a lot of resources and energy. And so if the cell doesn't need tryptophan, then it shouldn't make these enzymes to make tryptophan. So what can happen is that the tryptophan molecule, so this is the amino acid that I have drawn here in blue, it can actually bind to the repressor. And when it binds to the repressor, it kind of turns the repressor on. So it activates the repressor. And when it activates the repressor comes in and it binds to the operator. And when that happens, the RNA polymerase, even though it can still bind to the promoter, now when it reaches the operator, it gets stopped by the repressor because the repressor is now blocking the operator here. So to go down here and kind of finish our statement, it can be turned off by a repressor molecule, and we can add to it and say the repressor molecule is activated by the presence of tryptophan. So in conclusion, when tryptophan is present, the trip operon is turned off, which means that gene expression is going to stop. Now, this is only temporary because once that tryptophan is used up and goes away, it's going to release or unbind from the repressor. The repressor is going to remove itself from the operator, and then the RNA polymerase will be able to access the rest of the operon in order to transcribe those genes. So now let's take a look at the lactose or lac operon. And remember that the lac operon is an inducible operon. So as you can see from my simplistic drawing here, the operon has the same basic structures as the trip operon. So we still have our strand of DNA that has a region that codes for a regulatory gene, which codes for the production of the repressor. We have the promoter region, which is where the RNA polymerase will bind to. We have the operator region, which is where the repressor can bind to if it's present. And then we have the structural genes. And in this case, the structural genes are coding for the production of enzymes that are needed in order for the organism, in this case the bacterium, to digest lactose. Now, you'll notice that I went ahead and drew the repressor being attached to the operator. And that's because the default condition with the lac operon is a stop or a repressed operon. So the default is stop. And what this means is that gene expression is not occurring unless the repressor is removed and the operon is turned on. So in this case, the regulatory gene is always producing this repressor, and the default position is for the repressor to be bound to the operator. And what this means is that when the RNA polymerase binds to the promoter, it gets stopped. So it gets to the operator and it can't go any further. So it cannot transcribe these genes that are in the structural gene portion of the operon. Now, the thing that can remove the repressor is the presence of lactose. So if lactose is present, the lactose can bind to the repressor and it removes the repressor. So the repressor is going to leave the operator. And when it leaves the operator, the DNA polymerase is now able to travel along the entire operon and transcribe the structural genes that are further down on the operon. So the presence of lactose turns the operon on, or it induces it to start expressing the genes that are further along in the operon. And it does this by binding to the repressor, which causes it to be removed from the operator. Once the repressor is removed, the DNA polymerase is able to transcribe the genes in the operon.